Good afternoon to everybody. First of all, I would thank the organizer for the great opportunity they gave me to present this study in this context, in this amazing congress. This study that was carried out by the Molecular Anthropology Research Group of the University of Bologna. As molecular anthropologists, we are very interested in reconstructing the genetic structure of Italian population, mainly because due to its pivotal geographical position within the Mediterranean basin, the Italian Peninsula has long represented a natural hub for many human migration since the early Paleolithic colonization by Homo sapiens of the European continent. And also during the late glacial maximum, when most of the northern and central European regions were covered by ice, actually Italy represents one of the few refuge areas where human groups were forced to retreat and from which repeopled the world continent. And moreover, Italy played also an important role in the diffusion of agricultural techniques across Europe, mainly due to southern coastline population movements that brought the Neolithic culture to southern Italy considerably earlier than in other regions of Europe. In addition to these extensive Paleolithic and prehistoric population movements, a number of historical events have also played a role in reshuffling the patterns of Italian population genomic structure, and in particular the colonization of southern Italy by the Greeks, the huge expansion of the Roman Empire through the whole Mediterranean basin, and the occupation of Sicily by the Arabs. And according to this picture, we can hypothesize that a complex and heterogeneous genomic landscape could be observed in the overall Italian population distributed along the peninsula, despite its restricted geographical extension. However, up to date, very few studies have exploited genome-wide data to test this hypothesis. And in particular, these studies were mainly based on control samples recruited in the framework of large European genome-wide association studies, and thus not accurately selected according to, for example, stringent microgeographical criteria. To overcome this issue, we sampled around 800 healthy individuals belonging to four main Italian macro areas, namely Northern Italy, Central Italy, Southern Italy, and Sardinia, for which several studies have suggested that it could be found in this macro area a relative high internal historical and cultural homogeneity. And in particular, we selected 20 different provinces distributed along the peninsula in order to be representative of these macro areas. And for each provinces, individuals at least 3 generations native for a given provinces were selected. And DNA samples were typed with the Illumina core exome chip, that is a genotyping assay that allowed to test a wide selection of the most informative genome-wide SNP currently implemented in genome-wide genotyping array, a dense set of exomic markers, and also a select list of variants with no known clinical relevance. So first of all, we perform a descriptive analysis aimed at summarizing overall patterns of population structure, and in particular, we use principal component analysis to set the Italian variation within the context of the European and Mediterranean ones by comparison with 48 human groups from which available literature data, genome-wide overlapping literature data were available. And we can see that cluster of genetically related populations wrongly reflecting the genetic map of Europe could be identified, in particular with the first principal component describing a raw east-west axis of variation and enabling to perfectly distinguish peninsular Italians from Sardinians, while the second principal component instead described a latitudinal gradient of variation that was again perfectly evident along the Italian Peninsula. So to test the robustness of the identified Italian subpopulations, we compute membership probabilities for each individual to belong to a given cluster, and we average these probabilities for each sampling location, showing that provinces from northern Italy seems to be the less homogeneous one, with around 10% of individuals that could be actually assigned to another cluster, not the northern Italian cluster, but the central Italian cluster, for instance. While the percentages of misassigning individuals dropped to 5% in the central Italy and southern Italy, and to 3% in Sardinia, and being also restricted to a limited fraction of sampling locations. We also inferred the ancestry proportion for each examined genome, leading to the identification of 4 main ancestry components. For example, the red one is the ancestry component that is predominant in most Southern European populations, and it shows a uniform distribution along the Italian Peninsula, but interestingly, it reached near fixation in Sardinians, and it may be due to their long-term isolation, especially to Neolithic and post-Neolithic processes. And according to this ancestry component could be considered as a signature, as a relic signature of the early Paleolithic genomic background of Europeans. The red component instead, the red ancestry component, was typical of Middle Eastern population, being also evident in some Southern European population, and especially in Southern Italy. And along the peninsula, it followed a north to south decreasing gradient, and a perfectly similar pattern, also, even if less sharp, was evident also for the light blue component, which is instead characteristic of population from the Caucasus. And according to this, these 2 components could be considered as a signature of the early southern coastline population movements that mainly reach Italians from the Levant, from the Caucasus, and reach especially Sicily and southern Italy. And finally, the purple component was instead characteristic of Central Europeans and Eastern Europeans populations, and was represented in Italy with a north to south decreasing incline, and could be instead representative of the population movement characteristic of late Neolithic, and in particular the population movement that brought Neolithic culture into Italy from Central Europe and mainly the Balkans. So, well, having identified the actual presence of a cluster of genetically homogeneous provinces within the Italian population, we attempt to identify the genomic regions that drive this pattern of population structure. And for this purpose, we calculate FST index as a measure of genetic differentiation between each pairwise cluster comparison, and for each single SNP implemented in the genotyping array that we have produced. And in particular, we retain SNPs scoring in the top 0.1% of FST distribution within each frequency bin, and showing significant differences at the genome-wide level as regards their arrival in frequencies. And for instance, if we look at the results related to differentiation between northern and southern Italians, we have identified a larger genomic region spanning more than 1 megabase on chromosome 2 that contain 3 different genes that show unusual high differentiation with respect to genomic patterns. And in particular, 4 SNP drive this differentiation, being in low to moderate linkage disequilibrium with each other, and thus suggesting that we have captured at least 2 independent signals of differentiation. The first signal was related to a gene involved in neurotransmission, and in particular to two SNPs of these genes. One of them showed the ancestral allele being highly associated to high cholesterol levels and increase the risk of coronary artery disease, according to GWAS association studies. And a plausibly related signal emerged also from a gene involved in the processes of mRNA stabilization, and whose variants are known to be associated with increased level of triglycerides. A second, plausibly independent signal instead emerges from a gene involved in the transportation of vesicles in synapses, and in particular to one SNP whose ancestral allele was strongly associated with increased risk of type 2 diabetes. When we search for signature or natural selection on these loci, we found that the bulk of European populations have increased the frequency of the derived alleles, so not the alleles involved in increased susceptibility, but the complementary alleles of the derived alleles due to recent positive selection. While when we test the same signature of a selection in the Italian population, we fail to confirm this signature as regard the differentiation signal related to coronary artery disease, while we found a weak and barely significant signature of positive selection only in northern Italy as regards the signal related to the increased susceptibility of type 2 diabetes. So according to these results, we found completely different patterns of allele frequencies between the overall European populations and the overall Italian populations as regard loci that contributes to coronary artery disease and type 2 diabetes. In particular, we have found that the ancestral allele associated with increased disease susceptibility were overrepresented in the Italian population with respect to the other population, and especially were significantly overrepresented in southern Italy and in Sardinia with respect to the northern part of the peninsula. So according to the association of the risk allele, the ancestral risk allele, to high cholesterol level and insulin resistance, we can speculate that the complementary derived allele have increased their frequency in population of European ancestry due to positive selection in response to dietary-related pressure, okay? And for instance, population moving from the Levant and the Caucasus into the European continent are supposed to have forced to face progressively colder environments, and so probably have experienced, dietary shift towards high-calorie diets, high-fats diet, and according to this, the protective function, the putative protective function of the derived allele of these loci could have acted to counteract the side effects of these new dietary regimens. And in such a scenario, we can hypothesize that the selective pressure could have acted early along the continental migratory routes that move from the Levant to Western Europe, and in particular could have acted on the population, on the source population from which modern Western European population derived. And this may account for the differences in frequency between northern, Western European populations and Italy, since we have seen that the main contribution to the Italian genomic landscape comes from southern coastline population movements, and thus from populations that were not subjected to this selective pressure. So despite these results are obviously preliminary results, investigating pattern of population structure along the Italian peninsula promises to get new insight into the recent evolutionary events that mostly shape the Italian genomic background, and in particular into the events that made some Italian subpopulation more prone to develop specific disease in the context of the novel metabolic challenges imposed by modern lifestyles. So to complete this study, we plan to go on in identifying loss informative of population structure to replicate these SNPs on a larger cohort, and in particular to use this sample to select the most useful one to be used as controls in a genome-wide association study on type 2 diabetes and psoriatic arthritis. Thank you for your attention, and a special thank to all the people who are collaborating to this project in Italy. Thank you very much.